with AI. Oh, cool. Teacher was just being funny. And so were my with AI. Let's get that off. Um, hi. I'm just setting up the YouTube channel. Oh, your friend. I was going to say, what was that word? Also, for risk factors of obesity, I said diabetes, but I didn't specify. Will that be okay? So you didn't specify it was type 2 diabetes for the risk factors. It depends. Was this foundation paper or was this a higher paper? Hi, Addy. Give me 1 second. I am just setting up just now for the session. A higher triple. I think it depends what level that question is. It could be that that is not enough information and they would want you to actually specify that it was the type 2 diabetes. Would be potentially my assumption on that one. But the best thing is actually, do you know what? There's nothing you can do now. There's no point dwelling on the answers that you wrote. So the best thing you can do is just move on and focus on the next paper. One of the key things that students always do is they start talking about the paper, start talking about what they wrote, and then they get really stressed out because someone else will say they've written something else and that actually doesn't help you. So just move on, forget about it, don't worry too much. Hi, Chidra. Hi, Tony. Did you forget to say hi? I didn't even notice you come in and not say hi. Let me just put that in there. Right, do you want to go over the antibodies? I will go over antibodies, but I want to do, so I've had quite a lot of requests lately on what I think may come up, and I hate doing predictions. I don't really agree with predictions. However, I have been doing a lot of thinking, and I do want to support and help you guys as much as I can. So I thought I'd just jot a few things down. Um, um, Just note, Greg, to answer your question, you didn't write to see the additional page where you wrote some more. They will look and they will highlight for the examiner. So it should be okay. But next paper, make sure you do write it because that sometimes can get missed, but they should do it in the right way. Right. So do I think I think the grade boundaries will be the same this year. Um, um, okay. Oh, thank you. Thank you. That's lovely to hear. Right. I want to go through the just a quick one on a few of my thoughts. Now, these are not predictions. Okay. I'm not predicting because it depends on what day you ask me. So I went through again and looked at all my research and I keep changing my mind as to what I think that they are potentially going to be asking questions, but something in my gut is telling me specifically module 1, they'll ask you on anything. So that's just a given. You will be asked practical skills that will be assessed in that written exam. With module 2, now remember that comes up in paper 1, 2, and 3. That is going to be things like I feel quite strongly that lipids are coming up. Now, they can go in so many different ways with the lipids content because you've got the lipids as in just your fats, triglycerides, but you've also got the fact that they could look at the different types of whether they're monosaccharides, disaccharides. What am I doing? My brain. Let's try that again. Whether they are monosaturated or polyunsaturated. So they may go down that route. They'll also potentially look at the phospholipid and linking that to the biological membranes. Now, membranes come through everything. So these are all topic areas in module 2. Why it's in paper 1, 2, and 3 is because they can ask whichever way they want because they're the foundation parts of all the topic areas. But one of the things I would push a bit more on is make sure you're really confident on enzyme action and factors that are going to affect it. So they're either potentially give you a practical for you to design looking at factors affecting, or they are going to give you a graph and you're going to have to decipher and talk about what's going on. With cell division, I think that's going to be differentiation, but specifically for plants and plant tissue. So your xylem, your phloem, your guard cell, I feel like there's going to be a lot on that one. Now the paper 1 and 3 is obviously 1, 2, 3, and 5. So I feel like your insects are coming up. I feel like the insects exchange or insect transport potentially, but I feel more about insects exchange coming up rather than the animal exchange surfaces. But again, as I say, this is just where my gut is telling me. It's not a prediction as such. This is what I would say. Make sure you are solid in your revision for this. So heart dissection, they're going to give a dissection. They did. Your life is not eligible. What? That's so weird. Um, so I would definitely say that your heart dissection and cardiac cycle coming up, especially ECGs, actually, that would be something I have said. I do feel like ECG will come up and they could link that to neuronal communication because I feel like neuronal communication will be coming through whichever way they do it. Plant tissue, I do feel plant tissue is coming in. Xylem, phloem, specifically the translocation. If they're going to potentially ask you to plan an investigation, it could be on translocation rather than they did a lot on transpiration last year. So they may ask you about translocation and how you could do a practical based on that potentially. Neuronal communication, all of it. Hormonal communication, lipid soluble hormones. As I said before, I feel something strong about lipids. Muscles, absolutely, I feel that's coming up. And plant response, I feel like it would be more germination. Respiration, I feel like it could be practical respiration or anaerobic respiration. There wasn't that much on anaerobic respiration in the last couple of exams. For your paper 2 and 3, plant diseases, I feel like plants are going to come through quite a lot. So those of you who hate plants, I'm so sorry, but I do feel like the plants are coming through high this year. Biodiversity, I definitely feel there's going to be lots of different levels of biodiversity and plant adaptations for your classification and evolution. I feel like natural selection is coming in, but I've kind of put that in with patterns of inheritance because I feel like they'll go together. Gene regulation, transcription factors, and I do think that could be linked to the hormones in paper 3. So this is where those modules would come together. Hardy-Weinberg, I definitely feel like that's coming up with an inheritance question. How they'll do it, I'm not entirely sure though. GM applications and linking it to biotechnology potentially, batch and continuous fermentation, immobilized enzymes, animal cloning, somatic cell nuclear transfer, or actually gene therapy potentially in there would be what I would say. And ecosystems, carbon and nitrogen cycle, percentage energy efficiency, and managing ecosystems. I do feel like there's a big graph coming your way. So yeah, basically revise everything. That's one of the things that I would say, that whatever they decide to assess, it's got to cover the course, but it's about how they assess it and how they link it together. So yeah, pretty much basically revise as much as you can. Addy, have you actually seen my videos that I've already got on here specifically for population sustainability? Because although the specification has changed, all that's changed is that it has removed the case studies as they're not a necessity that you must know them. So I think teachers and students in the past have thought that you would only be assessed on those specific case studies, and that's not the case anymore. So the population sustainability side of things, that is literally just that they've removed bits rather than adding things in. It is a long topic. I do agree with you on that one. I feel like it's coming up. So the muscles for animal response, I feel like is huge. They've only asked it 1 year. And plant response, as I've said before, I feel like germination's coming up or something to do with gibberellins is definitely the bits that are coming up. But as I've said, if you're just coming across my YouTube channel now, what you should know is I have actually done videos on every single part of the course for you already in the playlist linked to the specification points. So that should help you. And it's specific to the 2025 exam. So the specification that came out in 2023 for this first exam in 2025. And the specification changes have more been about specifying what you need to know, what will be assessed, and removing things that were a bit ambiguous for both teachers and students. How do you revise module 1 and math skills? Validity questions are really interesting. So anytime you see a validity question, you always need to try to see what have they told you with regards to your sample size? What have they told you with regards to the standard deviation and error bars? How many repeats did they do? What was the uncertainty in the data they were collecting? So there's lots of different things, but those types of questions are very specific to the example that they've given you. So it's hard to just give a question like an answer to that without having an example to use, because they will give you any type of example. Whatever they decide is the thing they're going to assess this time round. So it could be for let me think. It could look at immobilized enzymes and be saying to you that they've done an investigation. They tested a alginate bead of an immobilized enzyme, and they were looking at how it effectively was effective at breaking down lactose into glucose and galactose, and they tested them at different temperatures. And so they were looking at whether they were free floating or whether they were immobilized enzymes. They were looking at temperature's effect on those enzymes, and they tested the temperatures at 5 degrees C, 15 degrees C, 40 degrees C, and 75 degrees C, let's just say. So we're looking for when it's really low, we're looking at when it's really high, we're trying to find the optimum in between. But there's such big gaps between those temperatures that is not going to give you a valid set of results. If you're planning that investigation, that's actually really bad because you've got too many gaps between your intervals, and so therefore you won't know exactly where the optimum is. So things like that, when it's talking about validity, should be about what's specifically in front of you. If they tested the carbon dioxide concentration in the atmosphere and they tested the temperature in the atmosphere, and they were looking at the correlation between the temperature and carbon dioxide concentration, so the carbon cycle, and how that's affecting the global warming concerns, then how many times did they test it? Did they test it every year? Did they test it every 5 years? Did they test it every 10 years? So that again would be about you looking at the validity. What was the sample size? What was the intervals between the testing points? Did they do a statistical test? Have they done enough repeats to work out a standard deviation? So any type of question like that, there are key words that you can throw in and just relate it to the example that they've given you. Okay. Right. So, are there any specific topic areas? So, you've said about plant and animal responses. So, they're quite, you are right, they're quite big topics. So, let me see if I can just pull up a question on them. Let me see. So, we want to look at, let's say, plants. So, this is quite an interesting one. If we look at this question, because somebody was asking about module 1 assessment, and you've asked about plants and plant responses. So here we're told that we've got some information about a student has access to a lab, and that access to lab is that they want to investigate the effects of the concentration of gibberellin on the rate of stem elongation in a pea plant. So they've given a specific example where it's a pea plant, and now what they're looking for is they're looking for you to tell them how you would outline the method that you would use. So what's the method that you would use? So I always say anytime we talk about this, you want to say, well, what's my independent variable? What's my dependent variable? And what are my control variables? Then I need to make sure that I do enough repeats to calculate my mean and standard deviation. And they specifically have asked about an appropriate statistical test. So I'm going to make sure that I graph out my results and do a stats test. So that's like my generic go-to answer if I was looking at a practical planning application. So in this case, when it's saying about me having to plan the method myself, how am I going to do it? I need to think about those words like validity. So therefore, what is my sample size going to be? So what is my sample size? How many repeats am I going to do? So you should know that you should have at least 5 repeats. You should also know that whatever your independent variable is, and this one is gibberellin concentration. So the concentration of my gibberellins, I should give them 5 values of that. And when I actually give those values, I need to try to be specific as to what are they going to be. So I want concentration of gibberellins, 5 values at least of that concentration. So am I doing a concentration of 10 to the minus 1? Am I doing a concentration of 1 parts per million? Am I doing a concentration of 100 parts per million? What is my values that I'm going to use for my independent variable? Oh, did you, Tony? How interesting. So then you say, well, what's my dependent variable? So what's the rate of the stem elongation going to be in this case? So if I'm going to measure the rate of the stem elongation, I need to think about, well, the rate is obviously the measured quantity over time. So I need to be specific in how I'm going to actually measure that. So my measurement will be the increase in length after a specific time period. So whenever you are doing your independent variable, make sure you've told them what you're actually measuring. Your concentration gibberellins, 5 values. My dependent variable, the rate, and I'm going to go by stem length. And obviously that stem length is going to be, because I'm looking at rate over time. So how much has it grown over the time to figure out the rate of stem elongation? And then what are my control variables? So always give 3 values because that will help you. And then I want a stats test, and they said to do the appropriate stats test. Now, if I've given 5 values of different concentrations, I therefore want to do Spearman's rank, correlation coefficient. However, if I decide that I want to compare the rate of stem elongation at 10 to the minus 1 and at 1 parts per million, then what I am going to do is I'm going to do an unpaired t-test. It is really, really important that you do not just name a statistical test if it is not the correct statistical test, because you'll lose marking points there. So if I'm using 2 groups, in this case, I've got 10 to the minus 1 parts per million and 1 parts per million. I'm therefore going to do that unpaired t-test. If I'm doing all 5 values, whatever those 5 values are, then I'm going to do my Spearman's rank correlation coefficient. So does that help with how to try to bring in some practical information and how to answer those questions? Here. Please could you go over the eyepiece graticule on microscopes? Please do check out my video on that one. That was actually a question in last year's paper. So if they're going to do it this year, it'll be a multiple choice question, I would think. I doubt they're going to ask the same style of question on that one. It will be more checking your understanding, but it could be a multiple choice question. So are there any other questions people have, or do you want me just to find another practice one to work through? Go for this one. If we look at this question there and work through this one. So whenever you get a question, one of the things I would say to you is make sure that what you're doing is taking the time to really dissect what it is that you're actually asking or being asked in the question. Oh, sorry, Jim, I totally forgot about antibodies. I will do that one. So I am doing that, Samira, with regards to the practicals and talking about what I think. So plant responses, I do feel like plant responses will come up, and I feel like the plant response that's going to come up could be linked more to the gibberellins, potentially to the germination. I have a feeling that that's coming, but I could be wrong on that one. And that's the thing. It's a case of, as I've said before, I change my mind every single time I do this because I'm like, oh yeah, that definitely is going to come up. And I'm like, oh, but actually they haven't done this. And that's the thing about the A level, that you can't really predict what is going to come up because they will change things so regularly. So it's about just making sure you know the content and therefore that you know how to answer the questions. So let's have a look at this one. And then after that, Chidima, I will go through for you antibodies. So animals and plants respond to changes. They're going to respond to changes in their environment. They're going to respond using chemicals known as plant hormones or plant growth regulators. A student carried out a two-part experiment to identify the contents of two unlabeled bottles. So we've got J and we've got K, which are unlabeled bottles. One bottle's got auxin, the other has got gibberellin. So therefore, straight away, I would say a question like this, what they're actually seeing is, do you know what happens with auxin? So do you know that auxin is for cell elongation? Do you know that gibberellin is for stem elongation? Gibberellin is also for the germination. Auxin is also for apical dominance. And auxin is involved in tropisms, such as your phototropism or your geotropism. So straight away, I'm already trying to prime myself as to what they might ask me in this question. I don't know what they're going to ask me. All I'm doing is I'm taking my time whilst going through this practical question to check whether or not I know my content. So then we say, right, in part one of the experiment, they had 30 seedlings. So what is that? That's my sample size. So I'm taking the time to note. The sample size is 30, which had their shoot tips removed. The 30 seedlings were then divided into 3 groups. So actually each of the 3 groups have got 10 and treated in a different way. So we've got no treatment applied. So that I know is my control. Why do I need a control? To compare what happens without anything being given. Then what you have is the next part. Solution J applied to the cut stem at the apex, and then K applied to the cut stem at the apex. So they've applied both of them, and they've applied them at the cut. So you're told here, treatments applied to groups 2 and 3. So that's what they're going to do. And then they're going to see, well, what happens? So then let's look at this. All the seedlings were exposed to light from all directions. So if it's coming from all directions, we would assume that there's no phototropism going to occur. So we would expect the growth to continue because we've been told the light is coming from all different directions, and they've been left for 7 days. Is this long enough? Would be one of the questions as I'm going through that I'm starting to think, do I think that's long enough? Are they going to get a good set of results from that? Then what are they telling us? In part to the experiment, what have they done? They've taken 30 coleoptiles. They've had their tips removed. Again, they were then divided into 3 groups, and they were given the exact same situation. So cut tip of the coleoptile rather than the cut tip, it was the shoot tips that were removed. In this case, it's the tip of the coleoptile that's been removed. So then it's asking me, okay, the coleoptiles of 4, 5, and 6 were exposed to light from one direction this time. One direction, so therefore I can expect there to be tropism occurring. Because the light is coming from this direction, I expect it to bend towards the light. Therefore, if I expect them to bend towards the light, I expect there to be cell elongation occurring on the shady side. So all I'm doing is I'm priming my brain. I'm thinking about what do I know about the different process and what can I then apply to this question. So the first question they're asking me to answer is, what are 3 variables that need to be actually controlled in this practical to ensure that they are going to get a valid set of results? So therefore, do I know what are all my other things that could affect the rate in which the growth and the elongation is going to happen. So basically, in this case, we're looking at anything to do with the likes of age, we're looking at the height or the length of the coleoptiles or the seedlings to begin with. What's the mass of them? Does that have an effect? What species are we using? So when we look at the variables that need to be controlled, it's all the things that could affect how they would interact with whatever this J and K actually is. So then they want you to note, I move on the next question. Group 104 were controls in the experiment. Explain why they were controls. Well, I already primed myself for that. So I primed that in the fact that I said, well, we're using controls, aren't we? So we need something to compare when there was no treatment whatsoever. What would the coleoptiles or the seedlings do on their own without any of the other things being given to them? So that's a typical question. And then what they'll do is they say, okay, now let's try and see if you can decipher what a set of results actually means. So this again is still module 1, but it's module 1 linking it to module 5 to the plants. So anytime you get given information in a table, in a graph, where you are going to have to pull the information out and actually work out a conclusion from the information, then that's also module 1. So they want you to work out what is in J and K based on the information that they've given you. So let's have a look at this. Thank you, Zoe. That's lovely. So let's have a look at this question then. And let's look at the results that they've given us so that we can try and decipher. So when we had, now remember, and this is where I would write little notes, 1 and 3 are my seedlings, and my 4 to 6 are my coleoptiles. So what did we have? Right, when there was no treatment whatsoever, there was an increase in stem length of 10 millimeters. So stem length was 10, and growth of lateral buds. When we look at J on the cut stem apex, there was no growth of lateral buds. Okay, no growth of lateral buds is making me think apical dominance. So I'm just going to write a little note to myself here, dominance, because apical dominance is where the auxin, when there is a high enough auxin concentration, that is therefore going to inhibit the growth of the lateral buds until the shoot gets far enough away to be able to get as much sunlight so that it can continue to grow, continue to photosynthesize, continue to make more cells. So therefore, this is where I'm thinking, right, apical dominance. So therefore, that tells me, I think, potentially at this first point, J might be auxin. Now remember, one of them is auxin, one of them is gibberellin. We're trying to figure out which one it is. So K, what happened to K? K was on the cut stem apex and it increased the stem length by 40 millimeters. Right, now remember, I've already said, because I've primed myself, that gibberellin was actually used for stem elongation. So this we can see is stem elongation. And because it's stem elongation and also not inhibiting the lateral bud growth, that again makes me think that this is gibberellin. So I've got some information. I've done 1 to 3. Let's double check 4 to 6 to see if they follow the same idea. Now, remember, we were having the light coming from a different direction, so we're expecting phototropism. With the phototropism, this is a live recording. So if you're asking a question, it is live right now. I am actually on Zoom, which you can join if you would like. At the moment, this is the third night actually I've gone live for you. I'm trying to do as much as I can to help you guys out. And I do it on my YouTube channel. So you think that it's recorded. I can reassure you, it is definitely not recorded. But thank you, Ninja. You're welcome. It's okay. It's not recorded. I don't see the point in recording. I don't plan these lessons. I'm literally just trying to help you guys out because I know not everyone can afford to pay for tutors and I want to support you. I mean, I've got 20 years of experience and if I can't give back at any point, then why do the job that I do? So yeah, that's the reason I do this, but also I go off in tangents sometimes. So I'm going to bring myself back to the question. Coleoptile, what have we got? Let's look here. We've got none. When there was no treatment, what we've got is vertical growth of the coleoptile, right? So that means it's going to go upright. It's going to keep going upright because there's vertical growth. When we put J on the coleoptile tip, the growth of the coleoptile was towards the light source. So remember, this is showing phototropism. Now, be sure when you write things like this, phototropism, that you're actually saying that it is positive phototropism. So positive phototropism because it's told you it's towards the light. So try not to lose marking points in your answers because you haven't been specific enough. It's not just phototropism. Phototropism is the growth which is directional to a stimulus is tropism. Photo, because of light, is that growth towards positive or away negative. You're from India, Ninja. Would you mind reacting to the NEET paper? It is an entrance exam for getting into medical college. Would you mind recording to the biology section of it? When you're asking me to react to it, what do you mean? As in you want to send it to me for me to have a look at it and then do videos on it? I'm not entirely sure what you're asking me on that one, but I'll come back to this question. When we applied K on the coleoptile, there was vertical growth. So the K didn't do anything because it was the same as 4. So 4 and 6 are exactly the same. So this one and this one. Yes, send me the link to Christine@aiqchat.com. This is the same result. So therefore, K has no effect when it comes to tropism. So when you're looking at this, use the information from the table to identify the contents and give reasons for your answer by taking your time to really dissect what it's shown you and also bring in your own knowledge. That's how you're going to maximize your grade. So that's the sort of questions they could ask you, linking it to module 1, which is about your evaluation skills. I'm analyzing this data. These are observations. Excuse me. So this is my quantitative and qualitative data that they have written down. So I am analyzing this data in a way that allows me to draw conclusions, and I'm using my science to back up why they are what they're or what's happening is the reason for it happening. Okay, Chidima, I promised you that I would do antibodies, so I'm now going to jump across to do a little bit on antibodies for you. Is it just the structure that you're struggling with, or is it that you are struggling with the different roles that they have to play? What part of it is it that's your main issue? If you're still here, Chidima, I don't know if you are still here. How do you advise revising for practicals? Zoe, how I would advise reacting for Chidima, I've just seen you join back on the TikTok. What is it that you're looking for the antibodies? So I'm about to go into it now. Zoe, to answer your question, how do you advise revising for the practicals? One of the things that I always say to my students is go to, she says, let me get it up right now. Is this one right? Here we go. So go to the specification. When you're on the specification, you want to look at this part. So this part tells you the key skills they're going to be assessing you on. So I like to use this page, which is 11. So this is page 11 of your specification. All I've done is I'm screenshotting it right now. So on page 11, what you can see here is these are the apparatus and techniques that you need to know. So they've already given you information. So to use appropriate apparatus to record a range of quantitative measurements. So therefore, they've already given you the quantitative measures you're going to have to do: a mass, the time, the volume, the temperature, the length, the pH. So I would say, how are you going to measure the mass? Do you know that you need to talk about using a balance? Do you know that you need to mention that you're going to zero or tare the balance to begin with before you start? Do you know that you're going to mention a weighing boat? And when you use that weighing boat, you're going to measure it without anything in it, so empty. And then you're going to measure it with whatever the solution is. So if you are looking at the practical part of the specification, these are the sorts of things that they will assess. Do you know what to measure your volume in, whether it's centimeters cubed, whether it's decimeters cubed? Do you know that we measure time in seconds or we measure time in minutes? Do you know that the length, you're going to use a ruler? If I'm using a ruler to measure my length, I'm therefore going to have a higher uncertainty than if I were to use, let's say, calipers, for example. So do you know how to choose the right equipment to measure specific volumes? If I'm using temperature, I'm going to use a water bath so that it is a thermoset temperature, and therefore I can ensure that it's maintained at the right temperature, and I'm using a thermometer to measure that. So these are the sorts of things that I would be going through, using the appropriate instrumentation to record the quantitative measurements, such as colorimeter or pedometer. Now, they actually put both of them in last year's paper. They did a lot on the pedometer when it came to the paper 3, I want to say, whereas with paper, she says, with the other one, colorimeter, hold on, I think that was an AS question. Let me just double check that. I may be wrong. It may have been A2 as well, but let me just double check. That's if I can spell. Yes, so AS paper last year had the colorimeter in it. A2 paper last year had the pedometer in it. So therefore, what you need to know is that they will be assessing these different practical skills, but they'll choose which one they want to do in different years, depending on what they've decided is that year's practical. So do you know both of them? So you need to make sure you can describe how to use a colorimeter, but also how to use a colorimeter for all the different tests. So students tend to memorize colorimeters, for example, with the glucose concentration, but do you know how to use the colorimeter to do the biuret test if you were looking at proteins? Do you know how to use the colorimeter if you were looking at the different saccharides and the different reducing sugars? Would there be a difference there? So do you know the steps of using a colorimeter? Do you know why you would use a colorimeter? Um, Moving on, as you can see here, C, zero dilution. So they did have a zero dilution question last year, but they tend to put zero dilution in every single year. So do you know? Yeah, I did go to Cambridge. I went to Cambridge for three things, actually. My postgrad for my teaching degree. I went for my a second postgrad on restorative approaches to conflicts in school, because I was a deputy of lower skill and then went on to be a director of student progress. And I also did my master's there, which was all about students' academic emotions and self-concept linked to learning. So yeah, I did go to Cambridge. You're absolutely right. And I still live in Cambridge. Haven't left since I came here 20 years ago. So for those of you who don't know, I'm from Scotland originally, and therefore I've only been here for the last 20 years, but I'm still here and I love it here. It is very nice. Using the light microscope. So they did do that one in last year's paper as well. So it's one of those things as you're going through, they are going to pick one of these and they're going to do a practical assessment on that. So it just depends which one they decide this year. Chromatography was a lot in the last years. I do feel that scientific drawing is going to come up, but linking it to dissections. I don't know why, but I feel like it's going to be your dissection of animal tissue because they did a dissection of plant tissue in last year's paper. You're doing BTEC and CSE. Oh, cool. Do you know what? It's a funny one. When you think about Cambridge, Cambridge and Oxford are the dream for you. I actually applied to Cambridge and Oxford for my teaching degree as a joke, which sounds ridiculous because I didn't go to university to do my undergrad until I was classed as a bit more of a mature student. I was 20 when I went. And was I 20? Or was I 20? Oh, now I'm trying to think. No, I was 22. I was 22 when I went. And so when I went, I was a mature student. But what I would say is that when I went to do my decision of where I was going for my postgrad for my teacher training, I wanted to come to England because they would pay me to train to be a teacher, whereas in Scotland, I would have had to pay for the tuition to learn to be able to do my postgrad. So that was the reason I came to England. And I applied to Cambridge and Oxford as a joke because I never thought for the life of me that I would get into Cambridge. And so the one thing I would say to you, as somebody who didn't complete their A level in biology, so I got a higher because I'm from Scotland. I've got the equivalent of an AS in biology. I have got a higher national certificate in leisure management, which has biology and business in it. And then, because I was a mature student, I then went to uni to do my biology. And then I got my BSc honours in biology before coming down here. And so it was a joke, thinking I'd be rejected. And actually, I didn't. I got in. So I love the fact that it's the dream. But remember, it's also about you trying and applying. Even when you think you might not get in, it's okay to be rejected. I got rejected from Oxford within a second. That was fine because actually it wasn't the right place for me to go and it wouldn't have been the right place for me. Whereas Cambridge, I got in and I got in because I'm a go-getter. I got in because I applied out of joke. They didn't tell me when my interview was. And so I phoned them up to ask them. And my interview was 2 days later. and I had to fly down that very day that I phoned them. And when I went for my interview, I literally just did this and I went blah, and I told them everything about stuff. And I was meant to prepare a presentation, and I just got up and I started speaking, and I started talking about what I was doing for my dissertation. And so therefore they could see that I was able to present in a way. I was able to link things together and find a way to make it work. And so they saw something in me. And because they saw something in me, they gave me the opportunity. And here I am now, 20 years later, trying to help other students find their belief that they can do it. Do you need to memorize the practicals for OCR? No. Shan, the answer to that question is no, because the required practicals are not required practicals. The practicals can be done in any which way your teacher wanted to do them. So for example, when I did the colorimeter, I did the colorimeter looking at glucose and calibration curves, whereas with some other students, they might have done it with a Biuret test. They might have done it with a different practical. With regards to the likes of thin layer chromatography, I did it with the photosynthetic pigments, whereas you might have done it with amino acids. So it really just depends. Also, as it says here, electrophoresis, I was very lucky in my previous skill. When I was head of biology, I linked with Amgen Bruce project, and we got to transform bacterial cells to take in a bacterial plasmid which had a fluorescent gene in it. And so I did electrophoresis with my students when I was teaching on the International Baccalaureate, and I had the opportunity. Now, when I went away from that back to A level and OCR, I didn't have access to electrophoresis kit. They're expensive. I couldn't get them on loan. And so therefore I couldn't do it. However, that doesn't mean you're not going to be examined on it. Just because my school doesn't have the equipment for it, doesn't mean you shouldn't know how to do it. So it's important you know all of these and that they can ask you about any of these. Do you have a list of command words for biology OCR? I would say with regards to biology OCR command words, you should really be using the OCR booklets specification. They have really good math skills booklet. They've got a practical skills booklet, and they also have a, what's the other one? Biological drawing skills booklet. You should be using them. And they're the ones that I would say to go to. I haven't made one myself because the way my brain works and the way that I do it, I kind of jump around and they don't work for me. And command words for me would just be me looking at a whole load of words that just jumble up and come together the same. So I don't work like that. My brain doesn't work like that. And I know that because I've learned a lot about learning. I've learned a lot about myself. And so I know that doesn't work for me. But there are people out there that have lists of command words. So what's different to concluding and evaluating? So concluding would be, for example, me saying the rate of reaction increased as the temperature increased. So there was a positive correlation up until the point of which the optimum was reached, and then the rate of reaction decreased. And then I would explain that in the sense that I would say, this is because of the kinetic energy. The kinetic energy, it's Shan, answer to your question. The neuromuscular junction doesn't have a video on its own. It should be in the playlist on animal response and in muscles. So it should be linked in there before I get to the bit, excuse me, I say, let me just double check that. YouTube video. Let me see what it's called. I think I called it muscle contraction. Let me just double check. So there's muscle contraction, skeletal muscles, and types of muscles. So it should be under the video, which is muscle contraction. Is that not on there, Shan? No, you're, because sometimes actually that does happen where I may have missed it off my playlist. So let me just quickly double check for you. Go to my channel. Playlist. Playlist. Where am I going? Animal response. Where are you? Where are you? Plant and animal response. Let me go in here. Oh, no, I don't want you to. What's that? Right, where are we? Skeletal muscle. Muscle contraction is in there. So is it not in the muscle contraction video? Yeah, it's in the muscle contraction video. Share, copy. Hold on, I'm coming back. I'm coming back. Coming back. Go to that video. So Sean, it should be in there towards the beginning of it. Right, so here back to you. So that was the concluding side of things. Now to go to the evaluating. So the evaluating part is where you're talking about they only did, although that's what the graph was showing, they only took readings at 5 different temperatures, and therefore I cannot be sure that the optimum temperature is, I don't know, let's say 37 degrees C, because they did 30 and they did 40 degrees C. So therefore, I would say that they would need to improve and do that again so that they can be sure what it is that they've actually got is the optimum. What else would I say? When it comes to evaluation, again, it all depends on what they've written in the practical side of things. But that would be the difference between concluding and evaluating. So evaluating is where you are scrutinizing about the practical, what went well, what didn't go well. If you did a statistical test, was there a significant difference between them? That would be as well another part of your concluding side of things. You're very welcome. Which A2 topics are on average worth the most mark? I couldn't tell you on that one. There isn't like a topic area specifically where you will get that topic area as being ranked higher than any other, because what they'll do is they'll hide the content they're assessing inside the topic that they're doing. So for example, if you're looking at respiration, really what you're looking at is not just respiration, you're looking at biological molecules, and you're also looking at biological membranes, and the way in which the molecules can be broken down requires enzymes and cofactors. So therefore that brings in your proteins and enzymes topic area, along with the fact that you need the movement of ions across a membrane. So that therefore also links the fact you're in the mitochondria, which is your cell structure. So it's all interlinked. It's not about A2 topics are worth the most marks. It's about how they'll assess it and what they're assessing. Concluding is drawing a conclusion that is you believing that you have found out an answer, and evaluating is supposed to be figuring out the answer. So yeah, you've got to be careful. Concluding, you're absolutely right. If you're drawing a conclusion, you're looking at the results and you're trying to make sense of what they actually mean. But evaluating it is about the science that backs it up and the validity of the results that you've got and how. So for example, if your results that you're concluding on have really large error bars, then the validity of your data is very low. So that would be the evaluation. My error bars are large. My standard deviation is large for that point and it's small for that other point. Tony, to answer your question about chewing gum allowed in the exam, I would say no. It's not allowed in the exam. However, if it is something that is written into a special educational needs plan, then it could be allowed, but that comes down to each individual person and each individual school and what they'll actually allow and tolerate. That's a weird noise. Right. Oh, see, but that's an interesting question. That was a hard question. I agree with you on that one, but you could see it coming a mile off. Bioinformatics and synthetic biology has been coming in, drip feeded in for the last couple of years, whereas this was a massive question that was asked specifically to assess students' knowledge, but it was actually broken down into three parts. So you were expected to know about bioinformatics, expected to know about computational biology, and then how that links to just genes in general and how DNA sequencing can be used to get those data to put in the database. Right, it has been a long day. Wednesdays are always my day where I get really tired because I teach a lot today. So I am going to go, guys. Thank you for being with me. Thank you for watching my videos. Please keep sharing, liking, commenting. If you have any questions, then please do ask on the videos because I do tend to reply. I will be back, if not Monday, I'll definitely be back on Tuesday. And it really depends if I'll be back on before then. But if not, then thank you, and I will see you when we come back.